In this video, we are going to explain what liquid biopsies are, how they are analyzed, and how they may be saving lives of cancer patients in the future. If you want to know how this works, stay tuned. The term biopsy is composed out of the Greek word bios, meaning life, and the word opsis, which is equivalent to a sight. Such a sight into life of a patient's unknown disease today is only possible through surgery. Surgeries, however, carry minor or even major risks potentially harming patients. Microinvasive liquid biopsies can diminish these risks for each individual. A popular way to do it is by looking at provided blood samples. With such an easy prepared sample, physicians can determine kinds and properties of cancers and are able to estimate the corresponding burden looking at cell-free nucleic acids floating in body fluids. Tumor formation process is elicited by mutations and accompanied by necrosis and cell death, which release nucleic acids into the bloodstream. Cancer cells can also actively secrete nucleic acids and send them throughout the body within vesicles. These cell-free nucleic acids can be isolated and scanned for mutations and abnormalities characterizing cancer diseases. But before all that, we must extract the DNA out of the blood, a process which starts with applying centrifugal force to any collected blood sample. In this first step, any blood sample from a cancer patient is mixed with an isotonic salt solution and some anticoagulating reagent to prevent coagulation. This mixture is spun inside a centrifuge to separate the heavier cells from the lighter parts of the blood sample. After spinning, you'll be finding 3 layers within your tube. A red layer at the bottom containing erythrocytes, a white layer in the middle where all the leukocytes are trapped, and a yellowish plasma layer on top containing proteins and nucleic acid fragments. This top layer is used to proceed with DNA isolation and purification.
In most cases, DNA is isolated from fresh plasma, applying a silica-based method. Therefore, we'll focus on explaining such a method, although other options exist. For silica-based methods, you'll need a hydrated silica matrix to start with. This matrix will then be dried by rinsing it with alcohols like ethanol or isopropanol. After drying, the negatively charged matrix needs to be prepared for adhering to DNA. Since DNA has a negative phosphate backbone and silica is negative as well, we need to add some chaotropic salts onto the matrix, providing cations to form electrostatic bridges between 2 negative molecules. As soon as the matrix is loaded with cations, our plasma is treated with proteinases to disrupt any remaining proteins and loaded onto the matrix where all negatively charged molecules adhere to the cationic salts. After adhering, the column is washed with ethanol to remove any impurities remaining before eluting the DNA from the matrix, applying deionized water. After isolation, DNA needs to be screened for mutations, revealing the nature of an underlying disease. At this step, the protocols vary to a great extent. While unknown diseases require costly full genome sequencing approaches, known disease sets can be identified using high-precision methods like digital droplet PCR to identify characteristic single nucleotide polymorphisms or similar known mutations. Here, we focus on explaining the targeted approach via digital droplet PCR. To reach high precision and sensitivity, any DNA sample gets distributed into small droplets either by hydrodynamic focusing, exploiting physical properties of oil-water surfaces, or by applying it onto solid-state microwell plates. Afterwards, every droplet or microwell is combined with PCR reagent containing primers for well-characterized mutations of a given disease. These primers are connected to quenched fluorophores, which start fluorescing as soon as the primer has found a binding site and is incorporated into a new transcript. By the time the PCR has finished, the droplets or microwells are screened for fluorescence, which is captured and quantified. The amount of fluorescence in a sample correlates with the amount of disease-carrying DNA and therefore allows identification of cancer-specific mutations.
At the end of downstream processed liquid biopsies, a disease subtype is specified and the underlying mutations are identified. Both identification and specification enable physicians to choose the perfect treatment for the identified cancer and the current progression of it. Nowadays, this technique is still under development and not yet applicable to be used in clinics on a daily basis of care. Despite the developmental status of liquid biopsies, it is likely that these techniques will soon evolve into spearheads of personalized medicine, helping to find cures and treatments for multiple kinds of cancers. However, this might still lay a long way down the road. Until then, we'll remain grateful for the work done by thousands of researchers all over the globe. And thank you for watching this video.